In this lecture, we're going to talk about fertilization and development. So just to review first, the egg cells are being produced in the ovary, and once the ovarian cycle begins at puberty, on average one egg cell per cycle will be ovulated into the uterine tube, often referred to as the fallopian tube. And it is in the fallopian tube that fertilization usually occurs. And if it does occur, then the fertilized egg or zygote will start moving along the fallopian tube towards the uterus. And we'll look, not in great depth, but we'll look at an overview of what happens to that fertilized egg. Also, I want to remind you about the ovarian and uterine cycles and how they're tied. So in the early part of the ovarian cycle, the main event going on is that under the influence of the hormone FSH from the anterior pituitary, several primordial follicles are recruited to become primary and then secondary follicles. And the main thing that's changing is it's not the egg itself, which is arrested in early meiosis, but the follicle cells that are surrounding it. Initially there's just one layer, then they start to divide in the primary follicle, and then in the secondary follicle you start to see cavities form within, and then usually just one secondary follicle is selected to become the mature Graafian follicle, and the cavities coalesce into a huge cavity inside. And the significance of the mature follicle, Graafian follicle, is that is the egg that's going to be ovulated that cycle. Okay, so that's sort of the first half of the ovarian cycle. The development of follicles, and the main hormone being made by the ovary is estrogen. Estrogen's made by the secondary follicles and by the mature follicle, not by the primordial and primary. And it is that estrogen that drives the proliferation of part of the endometrium, the inner lining of the uterus. It stimulates the cells to divide, and the endometrium actually thickens. They call that the proliferative phase. Okay, then the egg is ovulated into the fallopian tube, and what is left of the mature follicle is transformed into something called the corpus luteum, which means yellow body, and that the primary hormone driving that is LH, luteinizing hormone. So the corpus luteum begins to make progesterone, and also estrogen continues to be made. So now we have progesterone as well as estrogen being made in the ovary, and that brings about the secretory phase in the uterus, where glands, so these epithelial cells invaginating down in the endometrium, those are glands, and they're secreting things like nutrients in case the egg gets fertilized. So they call it the secretory phase. Glands are secreting. Okay, and then the corpus luteum degenerates as LH levels start to drop off towards the end of the cycle. So the corpus luteum, less and less progesterone and estrogen are made. Eventually the corpus luteum degenerates into something called the corpus albicans, white body. It's not making any hormones, and that built up endometrium starts to slough off, and that we call the menstrual phase, and that continues into the next few days of the next cycle. So if we go back to the beginning of this particular cycle, we see that when the ovary is in the early follicular phase, where there's no secondary or Graafian follicles to be making estrogen, without hormonal support, the endometrium built up in the previous cycle is going to slough off. So some cells die, there's a lot of blood loss because it's a very vascular layer, and you get the menstrual phase. So that's something we covered in our last lecture, but some of this information is going to be important. Especially I want to draw your attention to the to ovulation, which occurs about halfway through the ovarian cycle, and how once the egg is ovulated, within a few days, if you look down in the uterus, it's going to be in the secretory phase, and this is critical for successful pregnancy. The uterus must be in the secretory phase, so we'll talk more about that shortly. Here we see the egg that's going to be ovulated. So this is a Graafian follicle, sometimes also called tertiary follicle. So here we see the egg. They call it a primary oocyte. It is still not finished with meiosis. Right before ovulation, it did resume meiosis from prophase 1. It went a little further, but it's still arrested in meiosis. It hasn't finished. Around the egg that's going to go with it when it is ovulated is an acellular layer called the zona pellucida. Pellucida means clear zone. So that's that blue line going around the egg. And then we have some granulosa cells going around the egg cell, and that's called the corona radiata, the radiating crown. So all of that will go with the egg upon ovulation. And then the rest of the Graafian follicle that's left behind will become the corpus luteum, and that requires LH. Okay, ovulation is triggered by a surge of LH. They call it the LH surge. So here we see it. And now we've got the egg being released. Here we see in this picture the egg is being released into the fallopian tube. And notice, again, this is a review from our previous lecture, notice that the end of the fallopian tube has these fimbriae, these finger-like extensions. Those are going to start to undulate and wave and sweep the ovulated egg into the fallopian tube. So we're going to look next at fertilization, because usually fertilization occurs around here in the fallopian tube. Okay, now for fertilization we've also got to, of course, talk about sperm. So let's have a quick review of sperm. So they're produced in the testes, stored in the first of the 4 ducts, the epididymis, and sometimes they even complete their maturation in the epididymis. And that's where they stay until ejaculation, where rhythmic contractions of the ducts move some of the sperm, and we're talking about in the millions, some of the sperm into the long vas deferens or ductus deferens that loops up around the bladder, swings around, and merges with the duct from the seminal vesicle to form the ejaculatory duct, which comes through the prostate gland. That's where things get mixed with it, like fructose and fluid, various fluid, alkaline fluid, to make semen. And then from there the sperm passes through the urethra and out. And if we're talking about fertilization, of course that sperm is going to be released into the female reproductive tract, into the vagina. So the sperm need to get from the vagina up to the fallopian tubes. So they've got to, and this is when they start to use their flagella. Because remember, they're not using their flagella to move through the sperm ducts. They are moved by peristalsis there. But in the female reproductive tract, they've got to start swimming with their flagella up through the cervix of the uterus, up through the uterine cavity, and then they have to take one of the fallopian tubes, and usually just one's going to have the egg in it. So of the millions introduced into the female, only on the order of thousands make it to the fallopian tube where the egg resides. Okay, so once the sperm is introduced into the female, notice that it can't immediately fertilize the egg cell, even if it could be right there where the egg is within an hour. It still needs about 7 or 8 hours before it can fertilize the egg. We call this sperm capacitation. And what's behind this is that, if you recall, in the head of the sperm there was a sac of enzymes that the sperm need to get through this layer out here around the sperm. So this is the corona radiata out here, these cells. Here's the zona pellucida we were talking about, and then here's the egg. So to get to the egg, the sperm needs some enzymes to get through those other two barriers. And but that membrane, the acrosomal membrane, needs some time in the female reproductive system to become fragile enough to rupture and release the enzymes. So that's sperm capacitation. So let's look at that in this picture here. So here's a sperm arriving at the egg, and here's its acrosome with enzymes inside. So again, it takes about 7 hours before it can rupture. So here we see the acrosome reaction, where the enzymes are getting released, and then they help make a pathway through to the egg. Now in this picture it makes it appear as if one sperm can penetrate the entire barrier. It actually takes multiple sperm to erode a pathway through to the egg. So really it's the sperm that comes along at the right time that's where it's has enough enzymes to make it that last little bit through to the egg, and whichever sperm gets there first is the one that's going to be able to fertilize the egg cell. So this is the acrosome reaction that requires capacitation of the sperm. Notice, we don't really go into this much here, but it's interesting, is notice that the egg has sperm receptors that the sperm binds to. So here we see fertilization occurring. It's the fusion of the egg and the sperm cell, and once that happens, there's another important process that's shown here, and that is that the other sperm, which there's going to be many around the egg, the other sperm need to be kept out once the first sperm fuses with the egg, because you don't want multiple sperm bringing DNA, chromosomes, into the egg. So if you recall, the egg has 23 chromosomes. The sperm head, this purple thing in here, has the other 23 chromosomes in it. So, when they fuse, that will restore the correct diploid number of 46 chromosomes in humans. So you can't have more than that sperm. And so there's some measures that are in place that help prevent other sperm from getting in. We call it the block to polyspermy, too many sperm. So there's a block to prevent that. There's actually two main blocks. The first one you can't see in this picture, but they call it the fast block. And it happens when the first sperm head contacts these egg receptors, sperm receptors on the egg. It causes depolarization of the membrane. So some ion gates are going to open, and the membrane is going to depolarize, and that deters additional sperm from entering the egg. So they call it the fast block. So it happens very quickly. And then the so-called slow block, which is still pretty quick, but is called the cortical reaction. And what that involves are these vesicles right under the egg membrane, called cortical granules. So they contain calcium. And upon the first sperm fusing with the egg, these cortical granules, as you can see in this next store, these cortical granules release calcium into the space between the egg membrane and the zona pellucida. And so now we have calcium ions out here, and that also deters additional sperm from fusing with the egg. So those are the two blocks to polyspermy. Okay, and here we just see a scanning electron micrograph of a sperm head and the surface of the egg. So the sperm head is going to enter the egg, not the flagellum. Okay, I want to stop here for a moment and just talk briefly about timing here. The egg cell, once it's ovulated, has only 24 hours to be fertilized and then develop into a viable zygote and embryo. After that, it degenerates. So the egg's got 24 hours. Sperm, once they're introduced into the female reproductive system, are viable, meaning they can fertilize an egg for about 3 days. Okay, and that's an average. It's not precise, but about 3 days they can survive and be viable in the female reproductive tract. So if you put those two pieces of information together, you can kind of estimate, anyway, how many days in an ovarian cycle could introduction of the sperm into the female reproductive tract, i.e. intercourse, how many days could intercourse result in fertilization. So let's say the egg is ovulated on May 1st, and we won't get too picky with the times here. Just on May 1st, if it's ovulated, could fertilization occur if there's intercourse on the May 1st? Yes, that's a pretty obvious one. May 2nd, yes, at least part of it, because the egg is viable for 24 hours. So it could be the next day. Okay, but what about the day before the egg is ovulated? Could intercourse result in fertilization? Yes, because the sperm, even though the egg isn't even ovulated yet, they can be there waiting. They can survive, like we were saying, for about 3 days. So on April 30th, intercourse could result in fertilization. And the same is true for the 29th and even for part of the 28th. So that's a total of, from the 28th to the 2nd, that's about 5 days during that ovarian cycle that fertilization could occur after intercourse. So it's all timed with the egg. And people who are trying to have children, or even the reverse, trying not to, it's often helpful to know when, well, it is helpful to know when the egg is ovulated, because that's around the time that fertilization is possible. Okay, so here we see some sperm hovering around the egg, and we see fertilization going on here. Okay, once the egg is fertilized, that is when, we talked about this in an earlier lecture, that is when the egg finally finishes meiosis. Remember, it started way even before birth of the female that produced this egg. So that's a long time for meiosis to be completed. And really, it's only completed in eggs that get fertilized. So most eggs never complete meiosis. So anyway, that's what you see here going on in the egg. Hopefully this looks familiar to you. Remember meiosis. So this looks like anaphase going on here. Okay, so the egg finishes meiosis. The sperm is contributing half of the chromosomes, and here we see the very, very early zygote, the fertilized egg cell, where for a brief period of time there's actually two, they call them pronuclei. There's the one from the sperm and the one from the egg, each carrying 23 chromosomes, and they're soon going to fuse to form the zygote. Okay, now we don't go into too much detail about development, but we are going to look at an overview of it. So, first thing, big picture, is once fertilization occurs, development begins. You know, it's not going to stay as a single cell, and the first 2 weeks, so weeks 1 and 2, during that time, it's a pre-embryo. So it's not technically an embryo yet, but it's going to go through something we call the cleavage stage. So we'll look at that briefly in a moment. So that's what's going on in the first 2 weeks. And then week 3 through 8, this is really, in my opinion, the most exciting time of development. This is the embryonic period. So this is when you call it an embryo. So it's just a 6-week window of development. But so much happens during that period. That's when all the tissues form, all the organs form. So a lot going on. All the extra embryonic membranes that we'll briefly talk about, like the placenta and the amnion, they all form during the embryonic period. And then from the 9th week till birth, so that's up to the 9th month, it's the fetal stage. So we call it a fetus. Okay, so we're only looking at these stages very, very briefly. Alright, so here we see fertilization happening in the fallopian tube, and then the cleavage stage begins not too long after, so in about 30 hours later you get the first mitotic cell division. So see how we now have 2 cells. This is the 2-cell stage. And by the way, this is when sometimes those 2 cells separate, and each one develops into a separate baby, embryo and fetus, and that's how you get identical twins. When they're identical because they have the same DNA, because they came from the same zygote, which means it was the same egg and sperm chromosomes that formed that zygote. So anyway, we get the 2-cell stage, and then those are going to divide by mitosis to form the 4-cell, then 16, 32, 64, until you get like a little ball of cells. They call that the morula. And all the while, notice that it's slowly, the developing pre-embryo here is slowly being rolled along the fallopian tube. And by the time it gets to the uterus, it's called a blastocyst. And it's going to be in the uterine cavity for a few days, and then it's going to embed itself into the wall of the endometrium. That's called implantation. So I'm simplifying things a bit, but basically the first week of development is this cleavage stage, and I'll show you a picture of the blastocyst in a moment, this cleavage stage. And then the second week is the big event, is the blastocyst embeds itself in the endometrium, implantation, and then you enter the embryonic phase. Okay, when the blastocyst gets to the uterus better be in the secretory phase, or the pregnancy really will not be able to continue, because by the time it gets there, it's used up the stored nutrients for the most part that were in the egg, which, remember, that's why the egg is so huge, partly because it's got lots of nutrients stored in it. But after a week, a lot of that's gone. So these secretions here in the lumen of the uterus are very important for nourishing the blastocyst until something called the placenta forms, which will take over providing nutrients. We'll also look at that briefly. But for now, it's very important that the uterus is in the secretory phase. Now, if you recall, in the normal ovarian cycle, the secretory phase in the uterus only lasts a couple weeks, and then LH levels start to drop, and and remember what happens to the endometrium then? It gets sloughed off. The uterus goes into the menstrual phase. So it's critical that that be prevented. And that is something that does happen if two blastocysts, they get washed out with the menstrual phase if the secretory phase is not prolonged. Okay, so the main event that occurs to keep the corpus, to keep the secretory phase, to prolong it, is that the blastocyst itself is going to secrete a hormone. Here's the name of it. Human chorionic gonadotrophin is the name of it. Long name. HCG. This hormone acts similarly to LH. It keeps the corpus luteum going in the ovary for a few more weeks until the placenta is formed. So you need to keep the corpus luteum alive in the ovary because that's what's making the progesterone and the estrogen, and that's what's maintaining the secretory phase in the uterus. So HCG is often referred to as the hormone of pregnancy because it's not going to be around unless there's a blastocyst that starts making it. And in fact, this is the hormone used in pregnancy tests. They look for the presence of HCG. If it's there, then the woman is pregnant. Okay, this just shows the cleavage stage, and I wanted to show you the blastocyst down here at the bottom. So notice how during development, you start with pretty much a solid ball of cells. That's the morula, means little mulberry. But then notice in the blastocyst stage, there's a big cavity. It kind of looks like the graphene follicle, but so there's a big cavity in there. And then I wanted to point out that all the cells that you see in the blastocyst, each and every one is genetically identical. They're all derived by mitosis from the zygote, the original zygote. So they all have the same DNA. And I wanted to also mention that not every cell you see here is going to actually end up being part of the embryo. Just some of the cells in this area, this cell mass in here. They call it the embryoblast. This is going to form the embryo. But some of these other cells, like these cells out here labeled trophoblast, they're actually going to form part of the placenta. And also some of the cells in the blastocyst are going to form something called the amnion, and also something called the yolk sac. So those are all extra embryonic structures that help the embryo during development, but don't actually become part of the embryo. So we'll be looking at all of that briefly. Here we see a blastocyst, and you see the embryoblast. No, it's in blue. But again, keep in mind that all these cells have the same DNA in them. Anyway, here we see the blastocyst in the uterine cavity. So this white space out here would be the cavity of the uterus. And you see it's now up against the endometrium of the uterus. That's the inner lining. Out here would be the myometrium with all the smooth muscle, and then the serous membrane on the outside. Anyway, this is a blastocyst is getting ready to implant. Here we see it's implanted now in the endometrium of the uterus. So that's implantation. And you see that all the while it's changing. And let's look at a couple more pictures of that. Again, we don't go into this in too much detail. It's really fascinating material, embryology, but it's not something we have time for in our course. So I'm not going to go into all these terms out here, but just let's look at the picture. This is the blastocyst at about 9 days after fertilization, so still very young. In this picture we see the embryo at this point is just sort of a flattened, two-layered flattened disc. So right in here that I'm drawing the red ring around, that's going to be the embryo. And then we also see the beginnings of 3 extraembryonic membranes that I want you to know at their basic function anyway. The amnion, which is a cavity that's going to eventually form and envelop the embryo in a bag of fluid. So we'll see some later pictures of that. The yolk sac, which is this yellow structure. It's quite large. It contains nutrients, and it does help feed the embryo in the early stages of development, although it is going to shrink pretty quickly. So it's but they call it the yolk sac. In other animals, or like in frogs, for example, it's huge and remains throughout development as a major source of nutrition. But in humans, it's going to be the yolk sac is going to be replaced by the placenta, which is not developed yet at this stage. Okay, and then here's a little bit later. This is 3 weeks after fertilization. So now notice they're calling it an embryo because we're past the pre-embryonic stage, the first 2 weeks. The lumen of the uterus is out here, so now this implantation is over, and the embryo is completely embedded in the endometrium of the uterus. We see the amniotic cavity better here. It's full of fluid surrounding the embryo. Here we see the yolk sac, still rather large. And then out here on the edge, on the periphery, we see these structures called chorionic villi. You remember that term, villi, from the digestive system, finger-like extensions. So these are finger-like extensions of the outer layer of the blastocyst, and we call it the chorion. So it's like there's an amnion, yolk sac, chorionic villi, and notice they go all around this developing embryo. But soon we'll see that some of them degenerate and others grow, and the placenta is going to form from the sum of the chorionic villi. Also notice that the embryo is attached to the chorion by a cord-like structure, the umbilical cord, or umbilicus, and in it is an umbilical vein and 2 umbilical arteries. And there's a blood vessel connection between the embryo and these chorionic villi, soon to become the placenta. So here's a later view where we see the yolk sac is quite small now, so it's not really providing much nutrients. Although one thing the yolk sac does, one function of it, in addition to providing nutrients early on, is that the cells that are going to form the stem cells for gametes. In other words, the cells that in males can undergo meiosis to form sperm, or in females can undergo meiosis to form egg cells. They actually originate in the yolk sac for some reason. They originate in the yolk sac, and they have to migrate from the yolk sac into the embryo, into the developing ovaries or testes. So the yolk sac gives rise to the germ stem cells. They sometimes describe them as the stem cells that form the germ cells, the egg and sperm. Also, look how big the amnion is now, full of fluid. The function of the amnion is protection. It acts as a shock absorber for the embryo and later the fetus. It also provides freedom of movement, which is important because the development's going on. Like you can see, the arms and legs are starting to develop. So you can't have the embryo and fetus lying up against structures. You want it to be free floating, more or less. Also, it's the amnion provides a constant environment, so it's very controlled in terms of the temperature and composition. Also, it's been noted that the fetus drinks the amniotic fluid and voids into it. So presumably that has some important function in development. This, by the way, is the sac that they talk about often ruptures right before a woman goes into labor. They talk about the bag of waters or water breaking. They're talking about the amniotic fluid. Okay, so we see the yolk sac, the amnion, and then look at the chorion, the chorionic villi. Remember, they did go all around the embryo or fetus, the embryo, but now they're only in going part of the way around. Those are the chorionic villi that will be the placenta soon. While I have this picture up here, I wanted to mention something about genetic testing of before birth. So the older method for testing the baby's chromosomes to check for things like Down syndrome, to make sure the baby doesn't have an extra chromosome, or now we also have specific tests for specific genetic diseases like cystic fibrosis or Huntington's, chorea. So you can have you can do genetic testing on the baby, but of course you need their DNA. So the older method, which is still used quite a bit, is to remove some amniotic fluid, and in there will be some of the baby's skin cells that get sloughed off. So you can stick a needle through the abdomen into the amniotic sac. It will self-seal. Remove some fluid and then extract some of the baby's DNA from those skin cells. So they call that amniocentesis because it's using amniotic fluid. Then there is a newer, although it's been around for a number of years, a method called CVS sampling, chorionic villus sampling. They can do this earlier than amniocentesis, and it's done intravaginally. So they'll go up through the vagina and take a snip of a chorionic villus, and they'll use the DNA in there. Because even though it's not actually coming from the embryo per se, it's, remember, all from the same zygote, so the DNA is going to be the same. So you can get the DNA from the chorionic villus as well as the amnion. Okay, now here's a close-up of the placenta once it forms. So just to orient you, the baby would be over here. Here's the umbilical cord connecting the baby to the placenta, which is shown here. It has two parts to it. There's the fetal contribution to the placenta. That's the chorionic villus, villi, plural. So this tree-like structure that I'm indicating here with the arrow, that's a chorionic villus. So you can see it up close better. Notice it's got blood vessels in it. Okay, this is all coming from the baby, from the chorionic villi. So this is part of the placenta. The placenta being, you could say, the most important of the embryonic membranes. Although the amnion and yolk sac are important, the placenta has many vital roles. So here's the chorionic villus. Notice the blood vessels run from the chorionic villi through the umbilical cord to the baby. In case you can't tell, that's the baby there. Okay, and there's a, well, so that's the baby's contribution. And then for the mother's contribution to the placenta, well, this is the endometrium that we're in. And notice that during the development of the placenta, some of the endometrial tissue erodes, and you get like spaces in there. Spaces. They call it intervillous space because it's spaces between the chorionic villi. And then notice how the mother's blood vessels are running over to that space and emptying into it. So what is in these spaces around the villi is the mother's blood. The mother's blood is out there in this space. The baby's blood is inside the chorionic villi. They do not physically mix during pregnancy. They are separated by a two-cell barrier, actually. The baby's capillaries, the endothelium of the baby's capillaries, and then you can't tell from this picture, but the chorionic villus itself has a simple cuboidal epithelial wall. So if you want to leave the baby's bloodstream and go out into the mother's blood, you got to go through two cell layers: the capillary endothelium in the chorionic villus, and then the chorionic villus wall. And then same thing if you want to move things from the mother's blood into the baby's blood, it's the opposite direction. So we've seen this theme over and over in the lungs, in the digestive tract, in the kidney, this idea of a two-cell barrier. And so this is the placenta you're looking at. It's the mother's contribution is this part of the endometrium that's bathing the chorionic villi in blood, and it's not stagnant like a pond. It's, blood enters from the mother's arteries, and then it's also older blood's drained away through veins. So it's replenished, the blood here. And so while the baby's blood is going through blood vessels in the chorionic villus, it's exchanging materials with the mother's blood. And there are lots of things that are exchanged, so let's just run through the list. These are the main functions of the placenta. Well, nutrients are a major one. So, after the material in the egg is used up and the yolk sac is diminished, and the secretory glands in the uterus are not providing enough nutrients anymore, the placenta takes over as being the nutrient source. So nutrients in the mother's blood, like glucose and amino acids, can diffuse across the placenta into the baby's blood, and then that blood, nutrient-rich blood, will be carried back to the baby. Another thing exchanged is going to be oxygen. So oxygen will move from the mother's blood into the baby's blood. See, waste products are going to move in the opposite direction, from the baby into the placenta, into the mother's blood, and be carried away. Hormones. The placenta starts making estrogen and progesterone and other hormones. And so it has a major endocrine function. And once it starts producing those hormones, the ovaries basically shut down for the rest of pregnancy. The corpus luteum is no longer needed because the placenta has taken over that hormone production of estrogen and progesterone. And so we could say that it has an endocrine function, also an immune function, so there's some antibodies cross the placenta. So it has all these different functions: endocrine, digestive, urinary, respiratory, the gas exchange. Now there are, because things can cross the barrier, that does lead to some problems if the mother, for example, drinks too much alcohol, and a lot of because some things that you wouldn't want to cross the placental barrier can. So or drugs, and so that's how you have babies born addicted to drugs. So there are some undesirable things that can cross the placenta, like drugs, some viruses. So it's not a 100% foolproof barrier, only letting good things across. One more thing I wanted to mention while we're looking at this picture is notice the umbilical vein. There's one big umbilical vein in the umbilical cord and two smaller umbilical arteries. And notice that the umbilical vein is red. Okay, it's red because it is carrying oxygen-rich blood from the placenta to the baby. So it's oxygen-rich, so the hemoglobin has a red color. So it's like the pulmonary veins. Remember those? They were veins because they were bringing blood to the heart. That's what veins do. But they were oxygen-rich. So the umbilical vein is similar to that. And the arteries, which are taking blood out to the placenta, so they're carrying blood away from the embryo or fetus's heart, but they're oxygen poor. So they are arteries, but they're blue in this picture. Okay, well, to wrap up, I just have a few more pictures of embryos. Again, we don't really go into the morphological changes that occur, but I did mention that the embryonic stage is exciting because that's when all the tissues form, the cartilage and the bone and nervous tissue, etc., and all the organs. And in this picture, of course we're not looking inside, but you can see like the eye starting to form. You see where the heart is formed and it's actually beating at this point. You see the limbs developing, the arms and legs. Notice we do have a tail in the early embryo, which later is resorbed. So we're not born with it. And then here's a 52-day-old embryo, so it's starting to look more and more like a baby. You see the ear. Now you see where the nose is. You start to see fingers and toes. Here's the umbilical cord, which is quite chunky there. So the umbilical vein and arteries would be in there. Here's at 10 weeks. So notice there's the yolk sac. You see how small it is by 10 weeks. You can see the umbilical cord. We're going to look at just one more picture. This is our last picture. 14-week. You see the umbilical cord quite well here, and what you mainly see is the amnion, the amniotic sac, and now we're in the fetal stage, right at 14 weeks, and it really does look now like a human baby. You see the ear and the eye, and there's the mouth, and so on. So that completes our brief overview of fertilization and development.